Good. Well, thanks. And this is actually a really exciting day for Aduro because much of what I'll discuss today has actually been a result of a really very long collaboration with several investigators from UC Berkeley. And what I will really be addressing today is much of my talk will be related to some of the results that have been directly from collaborations around Listeria and the insights from this organism that have led us to the development of both vector and small molecule-based immunotherapy strategies. And so what's shown here is depicted as a very simplified view of the tumor microenvironment. And the tumor, of course, isn't just tumor cells, as we all know, but it has multiple support cells, the tumor stroma, which not only can provide, of course, nourishment to cancer cells, but can provide also physical barriers. And then, of course, there's a whole different set of cells, some of which are shown here, that infiltrate tumors to different degrees to either promote the tumor phenotype, like for example in the case of myeloid-derived suppressor cells, or you've also heard very nicely this morning how the CD8 T cell can be harnessed, and I guess right here the cytotoxic T lymphocyte can also be used to inhibit the progression of the tumor itself. And so I think it's pretty clear that there's an ongoing interaction between the tumor itself, of course, and the host immune system. And for example, if you look at tumors between patients with the same type of tumor, there's a huge variation in the level of infiltration. And then, of course, if you look at other types of cancers, they have a very low level of infiltration. And I think that Ira Melman will talk about that later today. And so, given, obviously, that cancer immunotherapy has now become a treatment paradigm, and understanding these differences between tumors and within tumor types is obviously, at a mechanistic level, is key in the context of developing effective immunotherapies. And I tried to get a different slide than everyone shows, but one theme that we heard this morning, of course, is that different cancer types have different levels of mutational burden. And there's been a couple elegant studies performed in the settings of both melanoma and non-small cell lung cancer, two malignancies, of course, that result from high environmental insults, and as a result have very high mutational burden. And one thing that was found by Nair Rizvi, and this was published last year, is that the response to immune checkpoint blockade, in this case PD-1 blockade, you actually can segregate responders according to their overall mutational burden. And both in this study and in the study with Judd Walchuk actually showed, in fact, that some of those responses are against mutated tumor antigens. But yet other cancers, like for example pancreas cancer, and this is one cancer that's kind of in the mid-range in terms of mutational burden, what we find is that there's absolutely this large phase 1 study that Jim talked about earlier. There's really essentially no response in that setting of pancreas cancer. And so why is that? And then as you also heard, colorectal cancer, so if you have a high microstability index and so more mutations due to defective mismatch repair, very responsive to checkpoint inhibitors. But those colorectal cancers that don't have mismatch repair, so-called MSS tumors, have no response, yet they still have a high mutational burden. And so, this is, another schematic slide that most people show, similar to this, but, the point is that as you increase in mutational burden, you end up generating an increased level of neoepitopes, and in the context of response to immune checkpoint blockade, that's seems to be directly correlated. But both in the context of immune checkpoint blockade, can you make those responses at a higher level? You've heard some approaches today. And then, of course, other malignancies where you have no response. Is there other ways that you can actually treat these patients and so they ultimately can become responsive to immune checkpoint blockade? So I won't belabor all these points because many of them have already been discussed this morning. But one thing that's absolutely clear, and in the words of Suzanne Topalian, checkpoint inhibition is rapidly becoming the common denominator of cancer therapy. So some of the questions that, that we have are checkpoint inhibitors alone sufficient for a durable anti-tumor response? And you've now heard some of the new strategy of combining rationally various immune checkpoint inhibitor blockade. But is priming of T-cell immunity against self-antigens, some of which was discussed this morning, and I'll talk about this more, or neoantigens, the so-called antigens that result from high mutational burden tumors, is this an essential component of immunotherapy? And then finally, what is the role of the tumor microenvironment in the context of either initiating or sustaining an effective immunotherapy? So our overall hypothesis is actually that effective immunotherapy requires a combination of modifying the tumor microenvironment and also in that context to prime tumor antigen-specific T-cell responses is depicted graphically here in combination, of course, given that it's a common denominator and very effective, as you've seen, is checkpoint inhibition. So we'll talk about two principal approaches that we've been actually taking, both at developing now in the clinic as well, which I'll discuss. Sort of two different approaches, one that we refer to as exogenous T-cell priming. And so we're using here live attenuated Listeria monocytogenes that express particular encoded antigens. And what I'll show you, in addition to it being a way to direct the immune system by specifically for the antigens that we've engineered it to encode, a major part of its activity in vivo, both in mice and in humans, we think, is due, in fact, because it does modulate the tumor microenvironment. And then a different approach is actually using a small molecule that's been engineered to withstand, that has enhanced properties for actually binding sting in the context of tumors, is actually in the context of the tumor to directly modify the tumor microenvironment and force the cross-presentation of a diverse set of antigens that we think will be unique to an individual's own relevant antigen repertoire. And so can't give a Listeria talk without showing this slide, and it depicts in something that Dan Portnoy showed many years ago, is how Listeria, an intracellular pathogen, grows and spreads through cells. And Listeria expresses a host of invasins known as internalins, and it expresses many of them that actually program the infection of particular somatic cell types or non-phagocytic cell types. And then it becomes trapped in an endosomal vacuole. And in the context of mammalian cells, it induces another genetic program that's regulated by a master transcription regulator known as PERF-A, and induces a host of virulence genes that allow it to grow intracellularly in the host. And in particular, in addition to encoding phospholipases C, there's another cytolysine known as Listeria lysine O that allows the Listeria to escape from the vacuole and enter the cytosol. And in the context of the cytosol, there's a high level of expression of another virulence protein known as ACT-A that is decorated on the pole of the bacterium, and it hijacks host cell actin and allows the bacterium via host actin motility to invade and invaginate the neighboring cell and go through this process again. And as you can see here, that Listeria remains sequestered from the extracellular milieu. And this is really why the correlates of protection in Listeria are entirely T-cell immunity. Or in the words of Dan Portnoy, this screams vaccine. And so in the mouse listeriosis model, there's some very old data that's been repeated in multiple different papers, is that you can infect mice with wild-type Listeria at sublethal doses. And then several years or several months later, really in the life of the mouse, you can then challenge them with lethal levels of wild-type Listeria, and they're completely protected against challenge. And again, protection is both CD4 and CD8 T-cell mediated. On the other hand, if you deplete or delete the ability of the bacterium to escape this endosomal vacuole, even though it can still prime T cells that can be boosted and proliferate upon challenge, those T cells are entirely functional. So it's really has to get out into the cytosol in order to elicit a functional T cell response. And this requirement for cytosolic entry for priming of functional T cells really informs the development of Listeria and based vaccines, and then both the small molecule immune modulators. Of course, you're not going to use wild-type Listeria as a vaccine vector. And when we actually looked at this slide, it made no sense to us in the context of stimulating an immune response. Obviously, that occurs eventually through uptake by dendritic cells and cross-priming. And what we're showing here is growth and spread through phagocytic cells. And we're also giving these bacterium, and it's done in the field as well, in mice intravenously. And what's known is that one of these internalins, called internalin B, is actually a ligand for the hepatocyte growth factor receptor that's expressed in all hepatocytes. And also, in the context of growth and then spread of the bacterium to neighboring cells, this also didn't seem to make sense to us in the context of what it would take to have an effective vaccine. So we rationalized that the combined deletion of these two virulence determinants would essentially focus the infection to antigen-presenting cells and then actually prevent, in the context of an organ architecture, its ultimate spread into neighboring non-phagocytic cells. And that's sort of depicted through fluorescent microscopy here, where we have cause cells that were engineered. The actin was engineered to be a fusion protein with GFP. And these cause cells were co-infected with two different Listeria, a wild-type Listeria that had been engineered to express blue fluorescent protein, and then wild-type Listeria that expresses RFP. And as you can see here, these beautiful with wild-type Listeria, these nice comet tails that is really is really the signature of a cytosolic infection, and you can see it kind of poking out of cells at different places. And then on the other hand, the Act-A internalin B Listeria, which we refer to as LAD, that stands for live attenuated double deleted. You'll hear me use that term frequently. These blue bugs just hang around the perinucleus. Why they actually hang out there, we're not really sure, but that's what we observe. And we published many years ago that combined, when you give these live attenuated double deleted Listeria to intravenously, you get a very rapid clearance phenotype from mice, and that's also correlated with a very significant reduction in the level of hepatocyte damage as shown on the right panel. And so what we want to show here now with data, and this is first shown graphically, that the rationale for using Listeria, and I'll show you a lot of evidence around this, is that, of course, it's been published that it triggers a very profound innate immune response and interacts with a number of different pathogen recognition and other danger receptors, and activates a number of innate immune effectors such as NK cells, macrophages, and generates a very high level of Th1 pro-inflammatory cytokines, which in the context of infected dendritic cell help to activate that dendritic cell. And we actually, and I'll go into this later, engineer these Listeria so that they express and secrete antigens into the cytosol so that it facilitates presentation along the class 1 pathway. And then ultimately the hope is that you get some tumor cell destruction. And this is a slide from our very first clinical trials many years ago, just taking non-recombinant live attenuated Listeria. It was a dose escalation study, and this was recently published, and just asking a very simple question: can you see activation of innate immunity in the context of the peripheral blood? And as you can see here, you see a transient increase of interferon gamma and IL-12 and an upregulation of activation markers of NK cells. Again, two important players, we believe, in the context of effective immunotherapy. So how do we express antigens? And this is something that's really Pete Lauer has dedicated much of his career to. And Listeria is a gram-positive organism with a very thick peptidoglycan wall, and it didn't really evolve to express and secrete mammalian tumor antigens. And it's actually secretion into the cytosol is essential for efficient presentation via the class 1 pathway. And what he's developed is a number of different methods for placing antigen expression cassette into defined loci on the bacterial chromosome. And what that essentially does is preserve the overall biology of the organism in the context of its intracellular growth. And importantly, the antigens themselves are actually expressed as fusion proteins with a signal peptide from ACT-A, which facilitates the secretion of the encoded antigen from the bacterium into the cytosol. But one of the important points is that we also drive the expression of antigens from the ACT-A promoter. And it turns out, and you saw the movie, that ACT-A is the highest induced bacterial protein in the context of the cytosol. So by taking advantage and exploiting that particular promoter, we're expressing and secreting antigens in the right compartment at the right time to facilitate antigen expression. And that's kind of shown here, where we express antigens. First, you see very nice expression levels in infected mouse dendritic cells in vitro. And then we ask the question, if we make a recombinant protein, a recombinant vaccine that expresses AH1, which is a self-antigen for endogenous retrovirus in BALB/c mice, that if we actually vaccinate those mice that are bearing CT26 tumors, that we induce a very robust, as you can see here, gamma interferon CD8 expressing CD8 T cells that are specific for AH1. And so moving on then, how can we use this in the context of various tumor models? And I'll show you a number of different approaches. And I want to start here, which is actually some old data that we published several years ago. And this really was set up the rationale for our first clinical trial, is, in the context of innate immunity, what can Listeria do to elicit a tumor-specific response? And so the way we asked this question was to actually just inject tumor cells in the splenic artery, and so it drains into the liver. And as you can see here in untreated mice, you see those livers are filled with tumors. On the other hand, if we take those same tumor-bearing mice and treat with just the non-recombinant Listeria, and this is looking at 3 days after 3 weeks after tumor implantation, you see in many of the mice just obviously a very reduced tumor burden, and that correlates to about 30 to 40% long-term survival. And what we showed here, that this is entirely due to the activation and recruitment of NK cells in the context of the liver. And if you deplete NK cells at the context of treatment with Listeria, you lose all efficacy. And those NK cells, through many mechanisms that have been discovered by David Rowley, go on to then, of course, result in an adaptive tumor-specific T cell response. So later, in these mice that are surviving, if we deplete CD8 T cells, then they are not protected against challenge. On the other hand, if we express in that previous slide, which I didn't say, and I'll say it very quickly, on the other hand, if we express antigen, and this is just AH1A5, and so that contains a modification in the epitope, so you get a better binding affinity to the T cell receptor. All the mice, as you can see here, survive after a single treatment with Listeria. But what about if we express the endogenous antigen in a 4T1 tumor model, which is also AH1, can be a restriction antigen? And so mice were treated here, as you can see, in 4 different ways, either with just a nonspecific antibody with PD-1 blockade alone, with Listeria expressing this AH1 alone. And in both of these cases, you see some reduction of tumor, but it's really, and this was shown earlier in different settings by Jim, that you see this really nice synergy by the combination of Listeria with PD-1 blockade. And so we've been spending a lot more time, and this is work from Wei-Wen Deng, looking at what is the composition in mouse of the tumor microenvironment in response to Listeria. And I'll show you a couple of the results related to this. And the way these experiments are done is after treatment with Listeria plus or minus PD-1 blockade, what is the composition of the immune tumor microenvironment? And as what you can see here, the Listeria-dependent increase in CD8 alpha dendritic cells in the context of the tumor microenvironment. And it's also interesting when you look at the T cell function in the tumor microenvironment itself. So this is a fairly immunogenic tumor, and so in untreated mice, just by tetramer analysis, you see a pretty high level of AH1-specific CD8 T cells. But if you actually look at the function of those T cells, what you can see by treatment with Listeria expressing the cognate antigen, that those T cells, there's a high percentage of T cells that acquire effector function, and that, of course, is Listeria dependent, as shown here. You don't see that with the PD-1 blockade. And the other really important point, of course, and that was addressed in the previous talk, is really what the role of regulatory T cells are. And one marker of a productive or tumor microenvironment that is productively responding to immunotherapy is a change in the CD8 T cell, effector T cell to Treg ratio, and you can see that Listeria does that as well. So let me give you an update of some of our ongoing clinical data and where we are, and I'll spend most of the time talking about a phase 2A study that combined two different modalities, CRS-207, which is actually the live attenuated double deleted platform expressing the tumor antigen known as mesothelin. So mesothelin is overexpressed in a number of solid tumors. This is really some seminal work done by Ira Pastan and Elizabeth Jaffe, showing that mesothelin, in addition to mesothelioma, is expressed at high level in pancreatic cancer, some non-small cell lung cancer, ovarian cancer, and various other solid tumors. And we conducted a phase 2A study combining two modalities, and I'll talk about what GVAX is on the next slide. We have another study. This was a very successful study, as you will see, in following this up in a larger study comparing the combination of 207 and GVAX to just Listeria alone to single-agent chemotherapy, and that's a trial known as Eclipse. It's a 300-patient-plus trial, and enrollment for that was just completed, and that will be reading out later this year. And then an ongoing study, which I won't talk about anymore, and this is also being led by Elizabeth Jaffe, is a combination of CRS-207 and GVAX with Nivo that's provided by BMS, and that's known as a Stellar study. And that's a two-arm study comparing the combination alone to this immunotherapy combination with anti-PD-1. And this study, at least in terms of interim analysis, will read out at the end of the year. So first of all, what is GVAX? And why are we using mesothelin? And I already gave you the reasons in the context of how it's expressed, but Elizabeth Jaffe published a paper about 10 years ago as a seminal paper that patients that were treated with GVAX. So what is GVAX? It's just allogeneic tumor cell lines, and the pancreas GVAX was made by Elizabeth Jaffe that have been engineered to express GM-CSF, which, as you know, is a potent recruiter and differentiator of dendritic cells. And those cells are irradiated prior to vaccination. So the context is this essentially is unbiased priming, but importantly, these are ostensibly shared antigens. And what Liz Jaffe showed many years ago, although the responses with just pancreas GVAX alone were relatively small, the patients that did respond and had clinical benefit, the one unifying theme is that they had CD8 T cells that were mesothelin specific. And it was really based on that rationale, and these cells also express high levels of mesothelin, that we developed Listeria expressing the cognate antigen mesothelin, as shown schematically here. And so this was a two-armed randomized trial, 2-to-1, and these were all patients that were metastatic pancreatic patients that had already failed conventional therapies, largely gemcitabine-based. And see, these are really second and third plus line patients. And they were given 1 of 2 therapies, again, randomized. It was a 93-patient study, 2-to-1. We're in Arm A, and this was also based on other preclinical data that I'm not showing you, combining 2 treatments of GVAX followed by 4 infusions of CRS-207. There's an interval of 3 weeks. And after a month, patients can continue, if they're not progressing, to receive additional immunotherapy cycle. And this was compared to just GVAX alone. And as we can see here, and the results were really quite striking, and these results actually resulted in us getting breakthrough status from the FDA. They're striking on 2 different levels. The first is, if you just look at median overall survival, and this is again in metastatic pancreatic cancer, where there was a significant increase in overall survival, shown here from 3.9 months to 6.1 months in patients treated with Arm A. This has a very good hazard ratio. What does that mean? So when the hazard ratio is at unity, that means it didn't do anything, and when it's significantly below 1,
you have more of a treatment effect. And as you can see, this is very significant. And the other observation is we saw kind of a so-called long-term tail, which Jim talked about earlier, related to CTLA-4, it's around 10%. And there were 7 patients, as shown here, that were on this therapy for a very long time, and 1 patient continues to be treated. And so what were the immune correlates? And I'll just show you some of this data. And one thing that was clear, and this was in both treatment groups, either arm A or arm B that just got GVAX alone, that patients that did better had an increase in the overall mesothelin CD8 T cells, and this was measured in the peripheral blood. But the other important, and actually patients that were on arm A, at least post the first cycle, had a significantly higher level of mesothelin-specific CD8 T cells, which is published here. The other important thing is that many of the epitopes on mesothelin have been mapped by Liz over the years, and what she was able to show is that those patients that actually survived longer had an increase in the epitopes on mesothelin that they recognized, showing a greater size of a T cell repertoire that was antigen-specific. And some more recent work is a collaboration with Lisa Cousins, and this is a little bit of a non sequitur, but we're also conducting a clinical trial in patients that have mesothelioma, which is obviously all patients with mesothelioma express very high levels of mesothelin. And this is actually a study that's being combined with standard of care chemotherapy in the front line. And so patients receive a couple of vaccinations with CRS-207, then chemotherapy, then CRS-207, and then it's repeated on a maintenance basis. And we first asked the question prior to chemotherapy, but after Listeria treatment, by doing biopsies in the lung in 3 patients as shown here, and multiplex IHC using a panel of both myeloid and lymphoid antibodies, what does the tumor microenvironment look like? And there was some really important takeaways, and you can see here one of the important findings of this study is that we see an infiltration of some of the really good players in the context of promoting an effective tumor-specific immune response, like, for example, Th1 polarized macrophages, NK cells, CTLs, and as well as dendritic cells. And some of that data comparing pre to post in these patients shown here, where you see an increase in CTLs, and then shown also in mice a better ratio between CD8 T cells and Tregs. So just to summarize this part in terms of safety and efficacy, and show you any of this data, but we've treated now over 300 patients with this platform, and some patients I think have had something like 50 infusions, and it shows a very favorable safety profile. And one of the real nice things, and one of the reasons that we started working on Listeria many years ago is Listeria doesn't care how it gets inside of APC. So one of the big problems, of course, with viral vectors is that they need to dock with a host cell, a cognate receptor, to be endocytosed and reveal the nucleic acids. And Werner Goebel showed many years ago via opsonization, Listeria still is taken up just as effectively in APCs, and through expression of LLO accesses the cytosol. And my reason for saying that is that after every infusion with Listeria, we still see this transient increase in cytokines as well as other innate immune effectors are being activated. We have staple insertion of multivalent antigen expression cassettes. I just showed you one example of a single antigen, but really the work of Pete Lauer, we can now express antigens that take up, for example, 250 kilodaltons of antigenic space. So it's really kind of a tour de force. And I think we have some nice and encouraging clinical data. I hopefully showed you that it induces a robust immune response, both on the context of innate immunity, but also antigen-specific adaptive T cell responses as well. And I talked to you about the Stellar study. So going back to some of the early observations, and this was an old unpublished study from the Portnoy lab, and we spent a lot of time talking about the requirement for Listeria to access the cytosol in order to elicit a functional T cell response. And one response, or one data point that Dan observed many years ago was that Listeria that escapes into the cytosol follows an interferon beta signature, and bugs that are trapped in the vacuole, even though they can still signal through MIT88 pathways, do not induce interferon. And so That led to some seminal work in the Vance in Portnoy labs that showed that bacteria that produce cyclic dinucleotides actually directly bind and engage a cytosolic receptor known as STING, stands for stimulator of interferon genes, that in response to binding its ligand actually activates the signature cytokine interferon beta through the signaling pathway here, affected by phosphorylation of ERF3. Also in the context of activating STING, you also induce the NF-kappa B pathway through a process that's poorly understood. And another pathway that's induced, but still at this point little studied, you get the JAK independent of the STAT6 pathway and perhaps some induction of important T-cell recruitment cytokines. And so anyway, that was shown that particular cyclic dinucleotides produced by bacteria directly engaged and activated STING. But STING was actually discovered as being the cytosolic receptor that made interferon in response to herpes virus infection, which is a DNA virus. And actually, how that signal was transduced to STING was not clear until James Chen discovered that the host expresses an enzyme known as CGAS, cyclic GMP-AMP synthetase, that in response to binding double-stranded DNA in the cytosol, undergoes a conformational change and actually makes a cyclic dinucleotide known as CGAMP, so cyclic GMP-AMP. And initially, what was missed in that seminal study was that the structure of the cyclic dinucleotide made by CGAS differs from the structure made by bacteria. And this was discovered by Russell Vance and Dinshaw Patel that showed that it was actually this so-called non-canonical linkage, which I'll show you on the next slide, binds and activates STING. So you can see that STING has evolved, that it can recognize different CDNs that have unique structures. And that's just really kind of shown here, is like the bacterial product has a non-canonical linkage that goes from 3' to 5' and 3' to 5'. And the change in the mixed linkage has this 2' linkage instead of the 3' linkage. So it's 2' to 5' and then 3' to 5'. And that results in a bent molecule, more of a cup structure, and it fits better into the STING binding pocket, and in response, showing from the apoform to the signaling complex, you get a more of a dramatic shift and a closure of this beta sheet lid for the signaling complex, although it's really not this trivial. Russell Vance published more recently that there's actually different forms of STING that can also signal in response to binding the cyclic dinucleotides. And so then we became very interested in this pathway, and the question is, well, what would we do with it? Is this really one of the key signaling molecules that makes Listeria elicit such effective T-cell immunity? And a few years ago, some work from Ken Murphy's lab actually showed that it's well known that the entry point of infection of Listeria is actually the CD8 alpha dendritic cells. And if you actually, by using genetic knockouts that prevent the development of CD8 alpha dendritic cells, you lose largely the ability of Listeria to prime a functional T cell response, as shown here. About the same time, Tom Gajewski, studying a model in mice, it found that just looking at the spontaneous regression or growth rates of mice, found that an interferon beta transcriptional profile, like in patients that have melanoma that respond to immune checkpoints, in the context of mouse tumors, what he found is that interferon beta signaling was critical in the context of controlling the rate of tumor growth. So preventing the paracrine signaling of interferon by knocking out the major type 1 interferon receptor, or knocking out CD8 alpha dendritic cells, completely or largely ablated the ability of T cells to mount a spontaneous tumor-specific T cell response. And so for us, this was like a real convergence of we have to try the experiment. So we knew that Listeria activated interferon by directly activating the STING pathway in the context of CD8 alpha DCs, and in the context of spontaneous tumor cell priming, CD8 alpha dendritic cells were the important point. But before we got there, we had to think about targeting human sting. And it turns out that there's 5 different human sting polymorphisms that exist in different levels in the population as shown here. And this is a result from the 1,000 Genomes Project. And so what we did was make a very large panel, which I'll go through on the next slide, of cyclic dinucleotide compounds that we asked different questions in terms of their modification and how that resulted in signaling. And Sarah McWhorter made a number of different cell lines in 293 T cells that have a defective sting that express the different human sting polymorphisms and then also mouse sting. And shown on the next slide, and I won't go through, of course, this is just showing a fraction of the compounds, looking at the ability, and this is just an interferon, a reporter assay, luciferase reporter assay, looking at the activation of a reporter expression in these cell lines expressing various sting alleles in response to stimulation with various compounds. And in the red is shown the cyclic di-AMP, the canonical CDN that's made by Listeria, and that's compared to a compound that I'll be referring now to 80US100. And this is shown a depiction of the crystal structure of 80US100. And it's comprised of 2 purine nucleotides that are adenosine, and I'll show you data around that and why. It has this non-canonical linkage, 2' to 5', 3' to 5'. And what we've done is substitute the non-bridging oxygens with sulfur, and this is a chiral center, and it's turned out that when the 2 sulfur atoms are in a so-called RR position, so they're above the compound or below, depending on how it's flipped, that turned out to have the best activity in the context of sting binding affinity uptake. And then, of course, the rationale for making this modification was to make these compounds resistant to host cell phosphodiesterases. We've also gone through a very large panel of human PBMCs looking at activation in response to 80US100 stimulation as a function of sting allelic complement. And what you can see here is most of the various combinations that can occur comparing various homozygous and heterozygous alleles. And one thing that we found after screening about 100 patients, and it exists 2% in the population, we found 2 donors that had the most refractory allele, so-called ref allele, or we call it the Barber allele. Somewhat ironically, this is the allele that was discovered when Glenn Barber first cloned sting. But as you can see, that 80US100 signals in human PBMCs bearing all those various sting alleles. And so again, the concept here is just by the direct intratumoral injection of 80US100 via activating the sting pathway and modulating the tumor microenvironment in the context of dendritic cells, which have already sampled their environment, but for many well-established reasons are defective. It's just forcing those dendritic cells to cross-present, an unbiased group of tumor antigens. And so I'll just walk you through some of the data, and much of it has been published already. And this first study is in the most aggressive B16 melanoma model, where we seed animals on the flank and wait until these are well established 14 days later. And what's shown here is treatment with either cyclic di-GMP, cyclic di-AMP, or 2 compounds derived from each of those molecules that have this dithiol modification. And what you can see here as a function of this dithiol modification, we see really pretty profound efficacy in the B16 model, which is very aggressive. And what's shown here is actually comparing those 2 compounds given at different dose levels, either cyclic di-GMP or the 80US100. And simply by treatment, this is just 3 different times, 3 days, we see long-term survival in about 50% of the mice. And sometimes we observe vitiligo, which is, as T-cell responses against melanoma differentiation antigens. But really interesting is that cyclic di-GMP, which is a much more inflammatory and induces a much larger acute response in the tumor microenvironment, it's something I'll address later. While those mice all get cured initially, what's very interesting, those mice don't go on to develop an adaptive T-cell response and frequently die of metastatic lesions. In this particular case, this mouse, as you can see, has a tumor in its snout. And it was, at least in my life, the first time I ever knew that the B16 model could metastasize. It's entirely essential to activate STING in the context of the tumor microenvironment. As you can see here in a different model, if we just give a subcutaneous injection and we don't directly activate dendritic cells in the tumor microenvironment, we lose all tumor efficacy. And what's really nice is that work that was done between Dan and Russell and J.D. Sauer developed this mouse as a golden ticket mouse that expresses a mutant STING. And in the context of these golden ticket mice that have a defective STING, we lose all activity, and this is shown in comparison to CpG. And then, on the other hand, if we look in the parental mice, you can see that the cyclic dinucleotides work very well in the context of an intact host or intact STING. And we all know what's important: can you elicit a systemic CD8 T cell response? And the way that this experiment was done is that mice were seeded both with a flank tumor and also given tumor cells intravenously, and just the flank tumor was injected, and the question was asked, what was the impact on the lung tumor nodule outgrowth? And as you can see here, if mice were given in the flank just salt water, there was no prevention of tumor outgrowth, and that wasn't the case in the context of cyclic di-AMP. So are these mice protected against challenge? And what we've done here, and this is in the 4T1 model, where AH1 seems to be a rejection antigen, again, by treatment with different dose levels of cyclic di-AMP, this is ADUS 100, again, you can see here in the primary treatment, all the tumors go away and they're all cured. But the question is, what was the T cell response against AH1, which is the endogenous antigen expressed by these tumor cells? And as you can see, there's a reverse correlation between dose level and the magnitude of the T cell response. And we know in other models, for example, at 1 and 5 micrograms, we would get a much higher CD8 T cell response. And so this is also correlated then when you do a tumor challenge. And so in these mice, once they're cured, they're then challenged. And what you can see here, again, is a reverse correlation between initial dose of CDN and protective immunity. And those mice that were actually, whose immune responses were primed at the lowest dose, had the best memory T cell responses. And so, what are some of the remaining questions that we have? And these are some of our, early data moving on. And so we know that STING is broadly expressed in multiple cell types in the TME. We know that the both host and the bone marrow responses are very important, and I won't be able to show all of that data. But how does the actual acute response, that is the injected tumor, how does that affect overall anti-tumor efficacy at distal sites? And STING activation, I think we've shown, and I showed here, induces a systemic T cell response. But are CD8 T cells the sole effector cell that's required for protection, long-term protection, and there are other mechanisms mediating anti-tumor immunity? And the other important takeaway is that it's clear that the STING-induced systemic immunity follows a bell-shaped curve. And one thing that we've noticed, and this is data that Kelsey Gautier has been leading, is that in response to treatment, not surprisingly, in the tumor with cyclic dinucleotides, and we see a massive influx of neutrophils into the tumor microenvironment. And if we prevent that influx by pre-treating with an antibody that depletes neutrophils, as you can see, we partially lose the overall response. This is a group of 8 mice, and so about half of the mice, had progressive tumors. What we know, and this has been published by others and by us as well, is that at least in terms of the acute response in the injected tumor, the TNF is a major player in the collapse of the tumor, and that's largely through targeting the tumor vasculature, both in the context of TNF and interferon beta. And to ask that question, I think, pretty elegantly, what we just tried is that we used Enbrel, which of course is one of the major drugs used to treat rheumatoid arthritis, and it functions by blocking TNF. And as you can see here, in the context of treatment with ADUS 100 and treatment with Enbrel, we again have a major impact on the acute response. On the other hand, if we deplete CD8 T cells in mice that are cleared prior to re-challenging them with tumor, we see a partial response. When we don't deplete CD8 T cells, all the mice reject tumors when they're challenged. When in our first data showing that when we do challenge the tumor, where we've first depleted CD8 T cells, they contribute, but obviously aren't the only player in the context of protective immunity. And using at lower dose levels, what we can find is a very nice synergy by combining ADUS 100 treatment with a checkpoint blockade, both in the context here in the, this is an MC38 model in the treated tumor, but we now have nice data showing in a systemic model when we actually just treat the one tumor and then ask the question whether or not in the combination with the immune checkpoint blockade, do we have better efficacy against the distal untreated tumor, the so-called abscopal effect. We have really nice data as well. So I won't go through all of these points other than to say I think it's pretty clear that in terms of the optimal dose, it follows a bell-shaped curve where it's defined in the primary tumor of no activity to complete activity, but somewhere along this dose range is really the optimal therapeutic index in terms of eliciting a functional CD8 T cell response. And I really want to point out that it's really not known, at least I'm not so sure it's very important for us to spend much time thinking too much about the acute response in the injected tumor because, B16 cells grow so rapidly, they have a dividing rate that's much more rapid than human tumors. And so really the focus of our efforts now is to ask different questions. How do we optimize the T cell response against a diverse repertoire of antigens? And that's really the focus of our work. And so this is just showing the co-crystal structure of ADUS 100 bound to wild type sting. You can see the closure of the lid. And I went through a lot of this data, but again, ADUS 100 is comprised of both a dithiol modification, cyclic di-AMP, which actually we've shown in the absence of any sort of formulation is taken up more readily. Obviously, sting is a cytosolic receptor, and that's a very important component of this therapy. So we recently just received clearance from the FDA to take this compound into the clinic, and we will be conducting a large phase 1 and phase 2 clinical trial in collaboration with Novartis. And this will be in patients that have cutaneously accessible tumor lesions. So this is melanoma, squamous cell cancer, Merkel cell cancer, cutaneous breast, head and neck cancer, and B-cell lymphoma in collaboration with Ron Levy. And it'll follow a dose escalation study in cohorts of about 4 to 5 patients in a phase 1 and 5 sites in the US, including at MD Anderson and at University of Chicago, along with our collaborator, Tom Gajewski. And then we will conduct a very large phase 2 study, about 50 patients in 2 different arms that are sorted between UV-induced tumors and non-UV-induced tumors. So, think back to my very first introductory slide. Again, one thing that's known in tumors that have, environmental insults, you have a higher mutational tumor burden, a hot tumor phenotype, and they're more responsive. And so we can actually ask if we can increase the response to PD-1, and then we'll also look at non-UV induced tumors, and then rapidly combine with immune checkpoint inhibitors. So just want to very quickly note a couple of people. There's many of people here, and I don't want to leave anyone out, but in particular, I want to note Sarah McWhorter, who leads the STING program, Pete Lauer, who leads the Listeria program. David Connie was really the mind behind multiple different modifications of the cyclic dinucleotides. It's now done by Chudi Nabaku. And then it's been really a very long and fruitful collaboration with Dan Portnoy, as you can see, starting from the very beginning with Listeria. And it was really the fundamental finding in the overexpression of interferon in the correlates that led us to the STING pathway. And then many investigators from Hopkins, notably Drew and Liz Jaffe. So thank you.
I think we're a little late, but time for a couple of questions. 2 questions. Maybe I'll ask a quick one then. I think as I understand it, the Listeria response is associated with a lot of apoptosis of immune cells, and it may even be a little immunosuppressive, although of course it's also immunoinducing. Do you think that tied into this effect of, say, high dose of CDNs? I think there's a relationship to those two observations?
Yeah, absolutely. I think what's happening, high doses of CDNs, and you have data that supports this, you have this massive exodus from the tumor, and it's a direct apoptosis of antigen-presenting cells.
Yeah, thank you for the interesting talk. I was just curious about as you mentioned, you are doing the local injection into the immune tumors like B16s with your STING molecules. I was also curious about what happens in case of the tumors which are not immunogenic tumors. Kind of thing.
Yeah, that's an important question, and one that we're going to ask clinically in the context of looking at UV-induced versus non-UV-induced tumors that don't have the same extent of inflammation in non-UV-induced tumors. We're actually right now beginning to set up different mouse systems that don't have that same level of inflammation. And of course, there's something I hear Ira Melman call an immune desert all the way to infiltration. Of course, there's a spectrum in between that. And one thing that we'll be testing in our clinical trial is really what is the level of inflammation prior to injection and following injection, both in the treated tumor and untreated tumor. And we'll, I think, begin to get some sense of, does activation of the STING pathway prevent a tumor microenvironment that's favorable to response?
I think we have time for only one more question back there.
Hey, Tom. Maybe I missed this, but I think you showed that your small molecule was STING dependent in its anti-melanoma effects. Did you look, was that type 1 interferon dependent?
We haven't looked at that directly. Good question. I think the question will be is really what the function of the CD8 T cells that we induce. My guess is that again the tumor itself is all TNF dependent, but it's really in the context of CD8 T cell signaling. I think the best data are probably from humans and activate the beta interferon transcriptional profile and T cell responses.
Okay, let's break for lunch back at 1:10. Thanks to the speakers.